Bismillahirrahmanirrahim. I am Hiddayatullah Khokhar, lecturer in Botany, Government Degree College, Mirpurkhas. Dear student, in last lecture we discussed about chapter number 6, Kingdom Prokaryotic. So, in continuation to that lecture, we are starting again from classification of bacteria on the basis of nutrition. In last lecture we discussed about photosynthetic bacteria and chemosynthetic bacteria. So today we discuss about heterotrophic bacteria. Heterotrophic bacteria are those which do not produce sugars from carbon and hydrogen for their survival and reproduction. It means that these bacteria are dependent on the other organisms. They are unable to synthesize their food themselves. So heterotrophic bacteria have been divided into three categories: saprotrophic bacteria, symbiotic bacteria, parasitic bacteria. So, saprotrophic bacteria are decomposers, putrefying bacteria. Saprotrophic bacteria get food from the dead organic matters, dead bodies of plants and animals. So, because these bacteria have a complex system of enzymes, with the help of these enzymes, they convert humus into simple compounds and then into energy. As we know that humus is a complex organic compound that is found within the soil, and this humus is synthesized, is formed by the partially decomposition of dead bodies of plants and animals. So humus always increases the fertility of soil. So saprotrophic bacteria have the potential of recycling of matter within the ecosystem because they are a free heterotroph. Saprotrophic bacteria may cause decay of the dead animals and plants. That's why they play an important role in cleaning the environment. So these bacteria may release key ions back to the environment. These ions are nitrates, sulfates, phosphates. So these bacteria may also cause souring of milk, cheese, butter and vinegar formation. So we take example of this class, Clostridium acetobutylicum. This bacteria always forms butyl alcohol from the simple carbohydrates. Similarly, Acetobacter oxidize organic compounds and oxidize organic compounds into lactic acid and vinegar. Similarly, Xanthomonas mobilis produces ethanol, sorbitol or gluconic acid from simple sugars. Now we discuss about symbiotic bacteria. Symbiotic bacteria are found in association with other living organisms. They obtain food from the host without harming to that host. It means they have the potential to live with other living organisms and they get benefit themselves and they also give benefit to their host. So these bacteria have the potential of nitrogen fixation. As you know that nitrogen fixation means nitrogen is present in atmosphere 78%, but that nitrogen is in inert form. Plants are unable to use that nitrogen. So bacteria are helpful in converting that nitrogen into nitrite and then nitrate. And nitrate is always used by the plants. So this whole process is done by nitrogen fixing bacteria. So we take example of Rhizobium radicicola, which is found in the leguminous plants roots. In pea plants we take example because they fix nitrogen within those roots. E. coli, which is found in human. Similarly, Azosporium species which are found in cereal grasses, they also fix nitrogen. Now we talk about the parasitic bacteria. Parasitic bacteria are those which depends on their host for their survival, for their food, and they give harm to their host. So far they are pathogenic. They may cause diseases, may cause death. These parasitic bacteria may be obligate, may be facultative. Obligate means these bacteria live only on the living organisms, while facultative means those organisms live on the living organisms. After the death of living organisms, they may live on their dead bodies. So far we are calling them facultative parasitic bacteria. Parasitic bacteria always obtain food by growing inside the tissue of host. And why they grow on the tissues of host? Because they lack certain complex system of enzymes. So far they depend on the host and they get food from their host. Similarly, they may cause many more diseases and they may also cause death of their host. We take example, Pneumococcus, Mycobacterium tuberculosis, Salmonella typhi. So now we discuss about classification of bacteria on the basis of staining. Staining, this procedure was basically used by Danish microbiologist Hans Christian Gram. So he classified bacteria into two main forms. One was the gram positive, other is the gram negative. Gram positive bacteria are those which retain the crystal violet color and they appear purple in color, while gram negative do not retain the crystal violet color and they appear pink in color. If we differentiate these two classes, main difference between the gram positive and gram negative bacteria is, let's suppose we have drawn here a chart. Few parameters are written here. Suppose cell wall. In gram positive bacteria, this is smooth and single, while in gram negative it is wavy and double layer. Cell wall thickness in gram positive, this is 20 to 80 nanometer, while in negative 8 to 10 nanometer. Peptidoglycan layer, this is thick layer in gram positive, thin layer in gram negative. Teichoic acid, this is present in gram positive, absent in gram negative. Outer membrane, this is absent in gram positive, present in gram negative. Mesosome, this is more prominent in gram positive, less prominent in gram negative. Morphology, cocci are spore forming rods, gram positive, while non-spore forming rods are gram negative bacteria. Toxin produced, exotoxins are produced by gram positive, endotoxin are exotoxin produced by gram negative bacteria. Lipopolysaccharides, this is absent in gram positive, present in gram negative. We take examples of gram positive bacteria and gram negative bacteria. As gram positive bacteria, Staphylococcus aureus, Streptococcus pneumonia, Mycobacterium tuberculosis, Bacillus cereus. So these are the gram positive bacteria, while gram negative bacteria are E. coli, Salmonella, Shigella and Pseudomonas. So this is all about the gram positive and gram negative. Thank you.